Have you ever stopped to think about how that piece of fruit you just ate actually powers your body? I mean, how does it turn into the energy you need to think, to move, even to just breathe? Well, the answer starts with a process called glycolysis. Today, we're going to break down this absolutely fundamental pathway, the universal first step in turning sugar into cellular fuel. And the star of our show is, of course, glucose. I mean, if glucose could talk, this is exactly what it would say. It's the primary fuel that literally sacrifices itself, breaks itself down to provide the energy that keeps everything alive, from the tiniest bacterium all the way up to our own complex bodies. So what's the official definition? Well, glycolysis is just a sequence of reactions that turns glucose into either pyruvate or lactate. But here's the most important part. The end goal is making ATP. You got to think of ATP as the cell's universal rechargeable battery. Glycolysis is the power plant that charges it up. And the fact that it's a universal pathway, that just tells you how ancient and absolutely essential this process is to all life on Earth. Okay, so that brings us to the big question for today's explainer. Why do we call this one single 10-step process the central hub of how we use carbs? Well, it's because, as we're about to find out, pretty much every road in sugar metabolism either leads to glycolysis or branches off from it. It's the grand central station of sugar. Ready to jump in? Great. Here's our game plan. We'll start with the basics, then get into the two main acts of this story. After that, we'll calculate the energy profits, see who's in control of the whole thing, and finally, we'll look at why all of this really matters in the world of medicine. All right, let's start at the very beginning. Glycolysis happens in the cytoplasm, the main fluid-filled space of every single one of your cells. It doesn't even need mitochondria to work. Now, this is absolutely critical for tissues like your red blood cells, which don't have any mitochondria at all, and especially for your brain. Your brain is a total glucose hog. It gobbles up about 120 grams of it every single day. To put that in perspective, that's about 60% of all the glucose your body uses when you're just sitting around. Now, here's a key point. Glycolysis can run under two very different conditions. When there's oxygen around, we call that aerobic glycolysis. The final product is a molecule called pyruvate, which then heads off to the mitochondria for some serious energy extraction. But when there's no oxygen, anaerobic glycolysis, the pathway ends with lactate instead. This is so important when you're doing strenuous exercise and your muscles are screaming for oxygen, or again, in tissues like red blood cells that can't use oxygen at all. So how do we make sense of these 10 steps? The easiest way is to think of it like a two-act play. Act 1 is the energy investment phase, and Act 2 is the energy payoff phase. It's a classic story of investment and return. You know, the cell has to spend a little bit of energy up front to make a whole lot more on the back end. This slide lays it out perfectly. In that first phase, the preparatory phase, the cell invests some of its precious ATP to kind of destabilize the glucose molecule, get it ready for what's next. Then in the second phase, the payoff phase, it reaps the rewards. And it's not just getting more ATP back, it's also capturing these high-energy electrons in a molecule called NADH, which, as we'll see, turns into a huge energy bonus later on. All right, let's unpack this journey. Now, don't worry, we're not going to get lost in the weeds of every single reaction. Instead, we're going to focus on the key, irreversible steps, the ones that act as the main control points for the entire pathway. So in phase 1, glucose gets a phosphate group added to it twice, and that costs 2 ATP. Step 1, using an enzyme called Hexokinase, is what traps the glucose inside the cell so it can't escape. But step 3, catalyzed by PFK1, that's the big one. This is the point of no return. Once the molecule gets its second phosphate group here, it is 100% committed to finishing glycolysis. There is no turning back. Finally, this now modified 6-carbon sugar gets split into 2 smaller 3-carbon molecules. And now for the good part, the payoff. Since we now have 2 of those 3-carbon molecules, everything that happens from here on out is doubled. We generate a grand total of 4 ATP and 2 NADH. This ATP is made through something called substrate-level phosphorylation, which is just a fancy way of saying a phosphate group is ripped directly off one molecule and handed to an ADP to make ATP. It's a really fast, direct way to make energy without needing any oxygen. And that final key step is step 10, catalyzed by pyruvate kinase, where the very last bit of ATP is generated. Okay, so let's do the final accounting. For anyone studying for an exam, this right here is the bottom line. What is the net energy gain from breaking down one single molecule of glucose? Let's walk through this table nice and slow. In phase 1, we invested 2 ATP, right? In phase 2, we generated 4 ATP directly. So, simple math, that's a net profit of 2 ATP right off the bat. But under aerobic conditions, we can't forget about those 2 NADH molecules. These guys get shuttled into the mitochondria where they fire up a process called oxidative phosphorylation. This is the main oxygen-hungry energy generator in the cell, and it makes a ton of ATP. In this case, those 2 little NADH molecules will generate another 6 ATP, bringing our total net gain to a very respectable 8 ATP per glucose molecule. But under anaerobic conditions, the story is completely different. Without oxygen, the NADH can't be used by the mitochondria. So instead, it's used to turn pyruvate into lactate. And what that means is we only get the ATP we made directly. So the net gain is just 2 ATP. This highlights a really crucial point. While anaerobic glycolysis is super fast, it is incredibly inefficient compared to the full aerobic process. It only extracts a tiny fraction of the total energy locked away in that glucose molecule. So a pathway this central and this powerful can't just be left running at full blast all the time, right? That would be chaos. The cell needs to tightly regulate it, turning it up when it needs energy and slowing it way down when it has plenty. This is what we call metabolic regulation. So how does the cell control the flow? Well, it's pretty smart about it. It focuses on those 3 irreversible point-of-no-return reactions we talked about earlier. The enzymes that carry out these steps, Hexokinase, the super important PFK1, and Pyruvate kinase, these are the key regulatory checkpoints for the whole highway. Now, what's really cool about this slide is just how logical all this control is. Take a look at PFK1. It's inhibited by ATP. I mean, that makes perfect sense. If the cell has a ton of its energy currency, it doesn't need to make any more. And on the flip side, it's activated by AMP, which is a big flashing sign that energy levels are low. It's just a beautiful example of feedback regulation, keeping the cell in perfect energy balance. So why does all this deep dive biochemistry actually matter in a clinical setting? Well, because when this fundamental pathway breaks down, the consequences for our health can be pretty severe. For example, let's talk about pyruvate kinase deficiency. Remember how we said red blood cells rely only on anaerobic glycolysis for their ATP? Well, if that very last enzyme, pyruvate kinase, is faulty, they just can't make enough energy. And without ATP to power their internal pumps, they can't maintain their shape and they literally burst. This is called hemolysis, and it leads to a specific type of anemia. Glycolysis is also front and center in cancer metabolism. There's this phenomenon known as the Warburg effect, where cancer cells basically become addicted to glucose. They run glycolysis at an incredibly high rate, even when there's plenty of oxygen around. They use this supercharged pathway not just for energy, but also to generate the raw building blocks they need for rapid, out-of-control growth. And finally, think about a medical emergency, like a heart attack. Tissues get starved of oxygen and they're forced into anaerobic glycolysis. They start churning out lactate, which then spills into the blood. If that lactate builds up faster than the liver can clear it, the blood's pH starts to drop, leading to a life-threatening condition called lactate acidosis. Phew. We have been on quite a journey through these 10 steps. So let's pull back for a second and just look at the big picture. What are the absolute must-know facts about glycolysis? Okay, here are the crucial points to lock in. First, glycolysis is the universal cytoplasmic pathway for breaking down glucose. Second, it has 2 very distinct phases, one where you invest energy and one where you get a payoff. Third, the net ATP yield is totally different depending on oxygen, just 2 ATP without it, but 8 ATP with it. Fourth, it's regulated at 3 key irreversible steps. And finally, and most importantly for clinical practice, problems with this pathway are directly linked to major conditions like anemia, cancer, and acidosis. And that leaves us with one final thought to chew on. We've seen how glycolysis is controlled inside the cell by its own products. But how does the body coordinate this pathway on a bigger scale? How do hormones like insulin, say after a big meal, or glucagon when you're fasting, reach into the cell to fine-tune this central hub and manage our body's entire energy budget? Well, that's a story for another explainer.